Chara sexual reproduction. Chara sexual reproduction done by the sexual organs. The sexual organs are two. They are globule and nucule. Globule is a male sexual organ of the Chara. Nucule is a female sexual organ of the Chara. These two sexual organs are developed at the node area of the main shoot. Male sexual organ is called globule. Female sexual organ is called nucule. These two sexual organs are developed at the node area of the main branch. Now we discussed about the development of globule. Development of globule. Globule belongs to male reproductive system of the Chara. See, it is developed on the node area. See, node area lateral side. Initially, one apical cell is developed. That apical cell is called globule initial apical cell. Globule initial apical cell. It participated in first transfer division to form two cells. Again, from the two cells, one cell participated in the transfer division to form three cells. Total three cells are present in a linear position. From these three cells, lower cell is act as a nodal cell. It cannot participate in the further divisions. And middle cell act as a pedicel cell. It is also cannot participate in the further divisions. But apical or upper cell is act as a antheridial mother cell. The antheridial mother cell is also called globule initial cell. It participated in the mitotic divisions to form two cells. The two cells again participated in the mitotic division to form four cells. The four cells again participated in the mitotic division to form 8 cells. 8 cells again participated in the mitotic division to form 16 cells. The 16 cells arranged in a two orals. In each oral, 8 cells are present. See, it is outer oral. In visible diagram, only 4 cells are visible, but the remaining 4 cells are present at the behind of these 4 cells. Total 8 cells are present at the peripheral region, and at center region, remaining 8 cells are present. The remaining 8 cells are again participated in the mitotic divisions. The remaining cells are again participated in the mitotic divisions to form 16 cells. At this stage, total 24 cells are present. The 24 cells are present in the 3 orals. In each oral, 8 cells are present. See, initially, only one apical cell, it participated in the mitotic division to form 2 cells. The 2 cells again participated in the mitotic division to form 4 cells. Again participated in the mitotic division to form 8 cells. 8 cells again participated in the mitotic division to form 16 cells. The 16 cells are arranged in a 2 orals. In each oral, 8 cells are present. Outer oral 8 cells take rest for few minutes, and inner oral 8 cells again participated in the mitotic division to form 16 cells. Total 24 cells are formed. From these 24 cells, 8 cells are present in outer oral, 8 cells are present in the middle oral, 8 cells are present at the center region. Total 24 cells arranged in a 3 orals. In each oral, 8 cells are present. From these 3 orals, outer oral 8 cells are called shield cells. They are developed into cover or shield of the globule. And middle oral 8 cells are act as a monobrial cells, and center oral 8 cells act as a primary capitulum cells. Outer oral 8 cells are called shield cells. Middle oral 8 cells are called monobrial cells. Center oral 8 cells are called primary capitulum cells. See, from these 24 cells, shield cells rapidly participated in the division. They develop into globule cover. The globule cover is also called shield. Initially, the shield cells are 8, but the shield cells are participated in the rapid division to form a cover-like structure around the globule. At the same time, middle oral 8 cells are elongated into tube-like structure. See children, here it is. These are the monobrial cells. Monobrial cells only 8. That 8 monobrial cells are elongated into tube-like structure. And center oral 8 cells. The center oral 8 cells are called primary capitulum cells. During this development, between the two monobrial cells, cavity-like structures are developed. In this cavity-like structure, ribbon-like structures are developed from the primary capitulum cells. The primary capitulum cells are produced ribbon-like structure. That ribbon-like structures are called secondary capitulum cells. The secondary capitulum cells are again produced ribbon, small ribbon-like structures. The small ribbon-like structures are called antheridial filament. Each antheridial filament look like a ribbon-like structure. It composed with more number of cells. In each cell, one haploid nucleus is present. The haploid nucleus is developed into male gamete. The male gamete is in spiral condition. The male gamete containing one haploid nucleus and 2 equal flagella. These are also called antherozoids. At matured stage of the globule, shield cells are broken and male gametes or antherozoids are comes out from the globule, and they are easily swim in water to reach nucule for the fertilization. It is the development of globule and development of male gametes in the Chara. The next point is development of nucule. Development of nucule. Nucule means it is the female reproductive organ of the Chara. The female reproductive organ of the Chara is called nucule. It developed at the node area above the globule initial cell. One initial cell is developed at the lateral side of the node area. That initial cell is called oogonial initial cell, or also called nucule initial cell. The nucule initial cell first participated in the transfer division to form 2 cells. From these 2 cells, one cell, lower cell, again participated in the transfer division to form 2 cells. Totally, at this stage, 3 cells are present. From these 3 cells, lower cell or basal cell act as a pedicel cell. It cannot participate in the further division. It act as a pedicel of the nucule. And middle cell is called nodal cell. It is also called tube cell. And above this nodal cell or above this tube cell, oogonial mother cell is present. See, oogonial mother cell also cannot participate in the further divisions. But middle cell, called nodal cell or tube cell, it participated in the mitotic division to form 6 cells. The 6 cells are arranged in a oral conditions. Center one cell is present. Around these one cell or around this central cell, remaining 5 tube cells are present. The tube cells are also called sheath initial cells. The sheath initial cells elongated into tube-like structure. They are spirally arranged around the oogonial mother cell. They are spirally arranged around the oogonial mother cell. The 5 sheath cells terminal parts are present at the oogonium. They are act as a cap-like structure. That cap-like structure part is called corona. Corona part. Corona means it is a cap-like structure present on the nucule of the Chara. And at the same time, oogonial mother cell elongated into large cell, it containing one egg cell and remaining cytoplasm, all cell organelles are present. Above these egg cell or oogonial cell, one receptive spot is present. Receptive spot mainly helps in the receiving of male gametes, And below these oogonial cell, central cell act as a stalk cell. Central cell again participated in the transfer division. It produced nodal central cell and stalk cell. Above these nodal central cell, stalk cell is present. Above the stalk cell, oogonium is present. It is the development of nucule of the Chara. Initially, one oogonial cell is developed at the node area. It developed above the globule. The oogonial initial cell participated in the transfer division to form two cells. From these two cells, lower cell again participated in the transfer division to form again two cells. At this stage, total 3 cells are present. From these 3 cells, lower cell act as a pedicel. It cannot participate in the further divisions. And the middle cell act as a nodal cell. It is also called sheath initial cells, also called tube initial cell. Above the nodal initial cell, terminal cell is present. The terminal cell is called oogonial mother cell. Oogonial mother cell also cannot participate in the further divisions. It directly develop into oogonium at the mature condition. But the middle cell participated in the mitotic division to form 6 cells. From these 6 cells, one cell present at the center region. It presented the lower part of the oogonial mother cell. It act as a central cell. The central cell surrounded by the 5 sheath cells. The 5 sheath cells are elongated into tube-like structures to form large tube-like structures. The tube-like structure cells, terminal parts are called corona cells. The corona cells act as a cap-like structure present at the nucule. And the tube cells spirally arranged around the oogonium. And the corona cells are present at the terminal part of the nucule. At the same time, central nodal cell, it present at the central, means basal part of the oogonial mother cell. At matured stage, the central cell again participated in the transfer division to form central nodal cell, and above the central nodal cell, one stalk cell. The stalk cell present at the lower part of the oogonium. Oogonium is a enlarged structure, it containing one haploid nucleus and cytoplasm. Above the oogonium or below the corona cells, receptive spot is present. The receptive spot mainly helps in receiving of male gamete. It is the development of nucule of Chara. During the fertilization, male gametes, spiral male gametes are received by the receptive spot and enter into the oogonium, fuse with egg nucleus of oogonium to form diploid zygote. The diploid zygote present in the oogonium. The diploid zygote development. That zygote is also called oospore. Development of zygote. Zygote is formed by the fusion of male and female gametes. Male gametes and female gametes are fertilized to form zygote. The diploid zygote initially take rest. Again, after some time, the diploid zygote participated in the meiotic division to form 4 haploid nuclei. From these 4 haploid nuclei, 3 haploid nuclei are degenerated or disappeared. Degenerated or disappeared. Only 1 haploid nucleus is developed into Chara plant. That developed haploid nucleus is also called lenticular cell. The lenticular cell participated in the division to form two unequal cells. That unequal cells are developed into rhizoidal initial cell, it is the small cell, and one large cell, protonymatal or protonymal cell. Protonymal cell develop into shoot of the Chara, rhizoidal initial cell develop into roots of the Chara, and these are developed into primary rhizoids, and above the protonymal develop into nodes and internode, finally develop into shoot, aerial shoot system of the Chara. It is the development of zygote in the Chara. We observed the life cycle. The life cycle always shows haploid condition except diploid zygote. The diploid zygote is take rest. After fertilization, diploid zygote is formed. The diploid zygote again participated in the meiosis division. It developed into haploid cells. Finally, we observed in the total life cycle of the Chara, anywhere the all cells are contains haploid nucleus except zygote. So this type of life cycles are called haploid life cycles. See, haploid life cycles present in the Chara. It is the complete life cycle of Chara. It belongs to chlorophyll C.